Welcome to this lecture on excretion in Annelida. Today, we are going to dive into the fascinating microscopic plumbing system of earthworms. If you have ever wondered how a creature without large kidneys manages to survive, filter waste, and maintain its internal water balance, you are in the right place. We will build our understanding from the ground up, looking at the biology, the mathematical models of excretion, and exactly how these tiny creatures thrive in their environments. Let us get started. Now that we know our topic, let us look at how the earthworm's body is organized. Imagine a bustling city where every single neighborhood has its own dedicated water filtration plant instead of one giant central dump. Because an earthworm's body is divided into many tiny rings or segments, it does not have a single pair of large kidneys like humans do. Instead, almost every single segment has its own set of microscopic plumbing tubes called nephridia. This decentralized system is incredibly efficient. It ensures that no matter how long the worm grows, every single part of its body stays perfectly clean and chemically balanced. Building on this idea of a decentralized system, let us examine the specific machinery at work. The most advanced type of nephridium found in earthworms is the meta-nephridium. Think of it as an open-ended tube connecting the worm's internal body cavity directly to the outside world. The process begins at a mouth-like ciliated funnel called the nephrostome, which actively sweeps in internal fluid. As this fluid travels through a highly coiled tubule, a surrounding network of blood capillaries reabsorbs essential salts, water, and nutrients back into the bloodstream. Finally, the concentrated toxic waste is expelled from the body through a tiny pore on the skin, known as the nephridiopore. To truly visualize what we just discussed, let us look at this detailed diagram of a metanephridium. On the left side, you can see the funnel-shaped nephrostome sitting in the coelomic fluid. Notice how the tube passes through the septum, which is the wall dividing the segments, and then becomes highly coiled. This coiled section is tightly wrapped in a dense capillary network, shown in red and blue, where vital reabsorption takes place. Finally, follow the tube down to the bottom right, where it terminates at the nephridiopore. This diagram perfectly captures the journey of fluid from the internal cavity to the outside environment. Having seen the physical structure, we must ask why this system is so critical for survival. The excretory system serves two main life-saving functions. First, as the worm digests food, highly toxic nitrogenous wastes rapidly build up. If these toxins were not continuously removed, the worm's cells would quickly be poisoned. Second, earthworms live in moist soil and constantly absorb water through their skin. Without a way to pump this excess water out, they would literally swell up and burst. By carefully controlling how much water is kept and how much is excreted, the nephridia act as microscopic bilge pumps, keeping the worm's internal pressure perfectly stable. Now that we understand the biological purpose, let us look at the mathematical model of physiological excretion. The total amount of waste that leaves the worm is not just the raw fluid sucked into the funnel. It is a calculated balance expressed by the mass balance equation, E equals F minus R plus S. Here, E represents the final rate of excretion. F stands for the filtration rate, the initial volume drawn in. R is the reabsorption rate, the crucial step where useful water and nutrients are pulled back into the blood. Finally, S represents the secretion rate, accounting for extra toxins actively pumped into the tube. If the worm is in a dry environment, the reabsorption variable R increases significantly, which in turn decreases the total excretion volume E. Before we move on, let us see this formula in action with a concrete example. Imagine a biologist measuring a single earthworm segment. We are given a filtration rate of 50 microliters per day. The capillaries reabsorb 40 microliters per day, and the blood secretes an extra 5 microliters of toxins per day. We need to find the final excretion rate. We start with our mass balance equation and substitute our given values. Following the order of operations, we first subtract the 40 microliters of saved fluid from the initial 50, leaving 10 microliters. Then we add the 5 microliters of secreted waste. This gives us a finite excretion rate of 15 microliters per day. This shows how efficiently the worm conserves water. Now that we have calculated the waste for a single unit, let us zoom out to the macroscopic scale of the entire earthworm. An earthworm does not just have one type of nephridium. It actually possesses 3 distinct types to maximize efficiency. First, there are pharyngeal nephridia, located near the front of the worm. Second, we have integumentary nephridia, which are tiny, forest-like structures scattered across the inner surface of the body wall. These discharge waste directly outside to keep the skin moist. Finally, there are septal nephridia, which are attached to the internal walls dividing the segments. These uniquely discharge waste into the intestine, allowing the worm to reabsorb even more water. To understand the vast scale of this plumbing network, we need a mathematical way to count these organs across the worm's body. Since nephridia are distributed across specific segments, we use a segmental counting formula. The total number, n, equals the end segment minus the start segment plus 1, all multiplied by the density d. We add 1 to ensure we are counting the segments inclusively. The variables are simple. S2 is the last segment in the range, S1 is the first segment, and D is the known density of nephridia per segment. We will use this formula to estimate the total excretory capacity of the worm. Let us apply this counting formula to find the total number of integumentary nephridia. Suppose we have an earthworm with exactly 100 segments. We're told these specific nephridia are located from segment 7 to segment 100, with a density of 250 per segment. We plug these numbers into our formula. First, we subtract 7 from 100 and add 1, which tells us there are exactly 94 segments containing these organs. Next, we multiply those 94 segments by the density of 250. This gives us a massive subtotal of 23,500 integumentary nephridia, discharging waste to keep the worm's skin moist. Having successfully calculated the integumentary population, we will now derive the next part of our total excretory capacity by looking at the septal nephridia. The mathematical reasoning is exactly the same. These organs are located from segment 15 to segment 100, with a density of 80 per segment. We set up our expression by subtracting 15 from 100 and adding 1. This inclusive addition reveals that exactly 86 segments house these organs. Multiplying these 86 segments by the density of 80 gives us our next subtotal, 6,880 septal nephridia. This chunk of our derivation sets us up perfectly to find the grand total. We have reached the final step of our derivation. To find the grand total of nephridia in this earthworm, we must sum the 3 distinct populations we just analyzed. The pharyngeal nephridia were given to us as a flat number of 1,200.
We add this to our calculated integumentary total of 23,500 and our septal total of 6,880. Adding these three values together yields a grand total of 31,580 nephridia. This massive final number mathematically proves our initial concept. The earthworm relies on a vast, decentralized network of over 30,000 microscopic units working in parallel.
We have covered a lot of ground today, so let us review the key takeaways. First, excretion in Annelida is a highly efficient, decentralized process driven by thousands of microscopic tubes called nephridia rather than a single centralized organ. Second, we learned that the actual volume of waste is governed by the mass balance equation, balancing filtration, reabsorption, and secretion. Third, this reabsorption step is absolutely critical for osmoregulation, preventing the worm from dehydrating or bursting. Finally, the combination of pharyngeal, integumentary, and septal nephridia ensures perfect internal fluid balance. You now have a complete understanding of how complex life adapts to manage its internal environment. Great job!